Chromosome mapping. Definition, types, importance. Part 2. UGC-NET Anthropology. Welcome to AnthroNet Academy, a study aligned with the UGC-NET Anthropology syllabus. Description box. Please visit the description box for books references. Dear learners, if you find this content helpful, please consider subscribing to AnthroNet Academy. Your support really means a lot and keeps me motivated to create more valuable content for you. Big surprise at 500 subs. Once AnthroNet Academy hits 500 subscribers, we're dropping something truly special just for you. A full video revealing 100 powerful, underrated, and must-read books list in Anthropology, complete with author names and insights important for UGC-NET exam. 2. Physical mapping. Physical mapping involves determining the precise locations of DNA sequences on chromosomes. It uses base pair as the unit of measurement. Physical maps provide a direct representation of the physical structure of a chromosome and the positions of genes along its DNA sequence. Markers used in physical mapping include ESTs, express sequence tags, STS, sequence tag site markers, and genome-wide DNA sequences. Methods of physical mapping. Cytogenetic mapping is a method used to locate genes on chromosomes by using their unique banding patterns. Each chromosome has its own distinctive pattern of light and dark bands. When observed under a microscope, these bands are numbered to help in the precise identification of specific regions within a chromosome. Cytogenetic maps can be created by correlating the position of a gene with the corresponding band on the chromosome. These maps are also called ideograms. Restriction mapping is a physical mapping method used to identify the positions of restriction sites on a DNA fragment. Restriction mapping involves comparing the sizes of DNA fragments created by two different restriction enzymes. Using RFLPs as markers for genetic mapping helps us to find the locations of certain variable restriction sites in a genome. However, most restriction sites are not variable, so RFLPs cannot be used to map many sites. Restriction mapping can be used as an alternative method in order to locate non-variable restriction sites. Fluorescence in situ hybridization, FISH, is a method used to visualize the position of specific DNA sequences on chromosomes. It involves labeling DNA probes and hybridizing them into intact chromosomes. In early versions of FISH, radioactive probes were used, but later fluorescent DNA labels were developed, which offer improved sensitivity and resolution. Sequence tag site, STS, mapping is a powerful mapping method for creating detailed physical maps of large genomes. It involves using short DNA sequences called STSs, typically 100 to 500 base pairs long, that occur uniquely in the genome. The map can be created by identifying the fragments that contain specific STSs using hybridization analysis or PCR. STSs can be obtained from various sources, including expressed sequence tags, ESTs, SSLPs, or random genomic sequences obtained through sequencing cloned genomic DNA. A commonly used marker for STS mapping is the EST, which is a unique sequence obtained by partially sequencing clones from a cDNA library. Radiation hybrid, RH, mapping is another physical mapping method that estimates the distance between genetic markers. RH mapping uses radiation to break DNA fragments. By controlling the radiation exposure, this method can create breaks between linked markers, resulting in a more detailed map. Rh mapping is valuable for locating various genetic markers and genomic fragments, particularly in regions with limited availability of highly polymorphic markers. Limitations of physical mapping. DNA fragments can be incorrectly mapped due to fragment breakage, deletion during replication, or contamination with host genetic material. There may be missing or incomplete coverage of DNA fragments in the mapping process, resulting in gaps. Restriction mapping method cannot be applied to large genomes. Physical mapping using fish is difficult to carry out, and the process of accumulating data is slow. In a single experiment, only a limited number of map positions can be obtained. To address these limitations and improve overall mapping accuracy, it is important to integrate and cross-reference both physical and genetic mapping. Importance of chromosome mapping. Chromosome mapping is important for understanding the organization and arrangement of genes on chromosomes. Chromosome mapping is important for identifying the precise positions of genes on chromosomes, which can help to understand gene interactions. Chromosome mapping is also useful to understand the structure and function of genes. Chromosome mapping provides information about the location of genes, which is important in the study of genetic disorders. Chromosome mapping is also useful for understanding evolution by comparing slight variations in gene arrangements among related species. Thank you for watching. Please like, share, and subscribe to my YouTube channel, AnthroNet Academy, for more insightful content on anthropology. Stay safe and connected.